Okay, why don't you stand over here? And,
Is it live?
yeah.
Okay. Hello, everyone. Thanks for attending this Friday seminar. I'd like to introduce Dr. Sean Gibbons. He received his bachelor's at University of Montana, where he studied molecular biology, microbiology, and French, and then got his master's of science at Uppsala University in Sweden, and later a PhD in biophysical sciences at the University of Chicago. He served as a postdoctoral scholar at the Center for Microbiome Informatics and Therapeutics at MIT. Currently, he is a Washington Research Foundation Distinguished Investigator and Assistant Professor at the Institute of System Biology in Seattle, Washington, and a Distinguished Affiliate Assistant Professor at the University of Washington, Department of Bioengineering and the Molecular Engineering Program, and a Data Science Fellow at the eScience Institute. So let's welcome Sean Gibbons, and I'm excited to hear his talk. Thank you.
Thanks, Caitlin, for that nice introduction. And it's an honor to be with you all today. These days I talk a lot to kind of medical audiences, so it's good to be back with ecologists and evolutionary biologists. I consider myself a microbial ecologist, even though I've kind of pivoted a bit into human health. And today I'm gonna think kind of from an ecological perspective on how do we leverage our inner ecology to treat a number of diseases that plague humanity. So one great thing I like about this field, about the microbiome field, is that it's philosophically altered how a lot of folks think about themselves, right? We're more than simply a bag of human cells. We contain a lot of other stuff. There's a lot of these other taxa that are very phylogenetically divergent from ourselves, that share our bodies. And to some extent, we've outsourced some of the functionality of our bodies to this ecosystem of alien taxa. Some organisms have done more of that than others, right? If you were a cow, you'll die pretty quick without your microbiome. You'll starve to death. A human could probably survive without their microbiome, honestly, like a mouse can. But we'd be weird and sick, and our organs would be malformed, and our immune systems would be messed up. So it's important, especially for kind of long-term health and longevity. And to give a kind of a little bit of a background on like how do we think of health or disease states in the microbiome? Ultimately, there's a lot of ways to have a healthy microbiome. I don't think there's any clear answer to this. One thing we've observed from time series analysis is that the median abundance of a given taxon is pretty conserved across humans. So this is a scatter plot showing the median abundance from time series from one individual and another individual, and you can see it's pretty correlated. Abundant organism in one person tends to be abundant in another person, and so also rare organisms or sort of low abundance organisms in one person are also lower abundance in another person. This is true across pretty much every pair of humans you look at, and sort of interestingly, another observation is that we're all kind of unique microbiome snowflakes. Right? Even though abundances are conserved across people, we all have these very unique fingerprints of what our microbiomes look like. So this is just a dimensional reduction plot on 2 dimensions of a bunch of time series, 10 different humans from a stool bank. This is actually people that were used for fecal transplants. They're donors that their material is used to give to people with C. diff infections. And so we had a kind of fossil record going back in time of all their poops. And if you sequence the time series, people look self-similar over time. They cluster with themselves. And so they're all kind of in their own little groupings. And what really distinguishes the differences between them is the absences. So any 2 individuals are sharing about 20 to 30% of the species in their microbiomes, and the rest is unique to those 2 pairs. If you look at 3 people, that drops to like 7 or 8% sharing, and then it gets lower and lower as you increase the number of people you're trying to get prevalence overlap across. When there are very few taxa at the species level that are present across like all humanity. And so, I think that's how you kind of square that circle, even though it looks like there's a conserved niche in a lot of people for a given taxon because its abundances are kind of conserved across people. The sort of set that you get, the functional set that you construct from when you're born to when you're now, is pretty unique to you as an individual. There's some stochasticity there, and there seems to be a lot of functional redundancy where people with very different sets still have a functional set. But what does a sick microbiome look like? This is some work I helped out with in my postdoc with a really talented grad student, Claire Duvalet, where we ran a meta-analysis across a lot of different diseases to ask whether or not there were any reproducible or consistent associations with disease across independent studies. And this is just showing a summary of some of those results. The sort of coloring here is showing things in red are enriched in the disease condition, and things in blue are enriched in the healthy controls. These are case control studies for, say, diarrhea, obesity, colorectal cancer, IBD, and HIV. And so we had at least 3 or 4 independent studies for each disease. And we're looking at organisms that are consistently enriched in at least 2 of these studies. And so you can find very different patterns across disease. One pattern we see is for colorectal cancer, you get a consistent enrichment in a sparse smattering of bad bugs, so to speak. Bugs that tend to be enriched in the disease state, like Fusobacterium and a few other kind of orally associated taxa. You see the exact opposite pattern in a disease like inflammatory bowel disease. There you actually see consistent depletion of the good guys, so to speak. And these tend to be butyrate-producing Clostridia. Butyrate is anti-inflammatory. People who have inflammatory bowel disease are often depleted in these good butyrate-producing organisms. So the loss of microbes can actually cause disease in addition to the gain of the wrong kinds of microbes. And other diseases are just like huge perturbations to the whole system and everything's turning over, like diarrhea, where you get a crazy number of hits. And the past 15 years of this field has been largely natural history. We're going out and it's like Alexander von Humboldt here going down to South America and looking at distribution of species along an altitude gradient on a mountain. We've gone around looking at different disease conditions and health conditions or demographic identities of individuals and seeing what's enriched where under what conditions. But like we talked about earlier, there's a lot of different ways to build these sets that are still functional, they're still healthy, they're still doing what they need to do for you. And mapping from an ecological composition to sort of functional space is a complicated problem. That I think the field's had a hard time getting towards. And so what I'll talk about today is sort of some approaches we're taking to move in that direction. And the central question from a healthcare standpoint, what the NIH cares about when it comes to ecology and the microbiome, is whether or not the microbiome is a mediator in phenotypic variation. Does it cause health or disease states? We know the genotype is one of these organismal filters. Variants in your genome can drive whether you get certain cancers or certain obesity or how tall you are and so on and so forth. And these things can all be modulated also by our environmental exposures throughout our lives, but the sort of genome is a filter upon those exposures. And what we're learning more and more, you see papers come out almost every day of examples where the microbiome is also one of these filters that seems to mediate response to either interventions or exposures or what have you. And the nice thing about the microbiome is it's a heck of a lot more plastic than the genome. It's a lot harder to CRISPR modify all your somatic cells than it is to putatively engineer the microbiome. It's still hard, but it seems to be more tractable. And I'm at a systems biology institute, so I'm a little biased, but I think we kind of have to take a systems scale approach. And inherently, that's an ecological perspective, I would say, where you take measurements from different layers of the system and integrate that information across those layers. The sum is greater, the whole is greater than the sum of the parts. And unless you look across the system holistically, you often miss what's going on. And so the ways we've tried to deal with this from a machine learning or statistical modeling framework to kind of discover new knowledge about this host microbiome interface is to do these deep phenotyping trials. This is just one example of the data inputs for a recent clinical trial that was run here at ISB in the Heath lab, where they were looking at COVID-19. This was early in the pandemic, recruiting patients and measuring kind of soup to nuts everything you can about them, right? You're sequencing their genomes, their blood metabolomes, their proteomes, single cell immune sequencing of their white blood cells, so on and so forth. Getting really, really resolved information about variability in the molecular phenotype of the host. And then our lab kind of came in as a way to kind of plug the microbiome into this picture. Even if you could predict the functional outputs of the microbiome, do those functional outputs affect the host phenotype? That's another layer of complexity that we don't quite understand how the mapping works there. And so, that's a lot of what we've done is looking at variation in the molecular phenotype of a host. What proportion of that variance is resonant with variability in the ecological dynamics, either cross-sectionally or longitudinally within the microbiome? And so this is a cartoon of like the general flavors of projects that we've worked on for the past 4.5 years, I guess I've been here. One is, like I said, the statistical modeling framework, where you take these deeply phenotyped cohorts that we have access to and try to learn out of those what you can. And I won't really talk so much about that today, except for one exception. And I'll focus today mostly actually on the other side of what we work on, which is mechanistic modeling. How do we build mechanistic models that can integrate this vast scale of complexity and make functional predictions for these really functionally redundant sets of taxa. And it'll be a particular genome-scale metabolic modeling and flux balance analysis that I'll talk about, with a bit of experimental validation for how we kind of validate those predictions. So, I think there's a lot of axes where the microbiome mechanistically affects host phenotype, but probably one is definitely like antigens and proteins in these microbes and how they're interfacing with the host immune system, which I won't talk about today. And I think the other big one is the production of small molecules. The microbiome is a little pharmaceutical plant sitting in your gut, and it's constantly transforming dietary inputs and host metabolites into a wide array of bioactive molecules, many of which are getting absorbed into the bloodstream and circulating throughout the body, touching all of our organs. There's this intimate conversation occurring between our microbiota and our bodies. We don't have a Rosetta Stone yet to understand what's being said, but that's kind of what we're working towards. And this is what I talked about earlier. Like, I'll talk a little bit about a kind of statistical learning project we've recently wrapped up, where it's relevant to this question of, is microbial metabolomics relevant to human health? In that we're trying to really partition the variation in the human blood metabolome into what amount of variance on a metabolite by metabolite level is explained by the genome, and what amount is explained by variation in the gut microbiome. And so this is work carried out by Christian Diener, a senior research scientist in my group, and Cheng Dai, who's in a collaborator's lab, where we had about 1,500 people with whole genome sequencing on the host side, along with detailed information on sort of demographic and background of all the individuals. So we could do a lot of confounder controlling, like BMI, sex, age, and microbiomes, in addition to blood metabolomes. So we had these three major data types all together for these 1,500 folks. We had about 1,000 metabolites we could robustly quantify across this 1,500 person cohort, 930. And we wanted to know for each one of those metabolites, are they associated with the genome and the microbiome? How much of the variance can be explained by either factor? So we had a kind of training set where we calculated significance, about 1,000 person set, then built these models and calculated R squareds for variance explained on a validation set that was not a part of the training set, to get kind of robust R squared estimates. And just to give an overview of some of those results, right? We see there's a ton of stuff that's just associated with the microbiome in green. We see stuff just associated with the genome in blue. And then there's a fair number of metabolites in the blood that are hybrid, that their variation is explained in part by both the genome and the microbiome. And this is the training and the validation sets, and the R squareds are pretty robust and correlated across all of these things. About 60% of all the metabolites circulating in the human bloodstream were significantly associated with either the genome or the microbiome in this 1,500 person cohort. And of that 60%, 69% of those were exclusively associated with the microbiome. So the majority of these associations with the blood metabolome are seemingly driven by the microbiome, and a minority are driven by the genome, at least within the context of the variation we see in this cohort. About the same number of hybrid models as genetics only models. And we had another paper that was published a couple of years ago, Barr et al. Did a kind of similar analysis, and we were able to show that the sort of R squareds they observe in this Israeli cohort, a totally separate analysis with different kind of data types even, are really conserved. So we see pretty good matching between genetics, especially, and the microbiome. We see some outliers here, but they seem to be related to the fact that I think that in this study they were using serum, and in our study we were using plasma. So we had some sort of different processing of the blood, essentially. Okay. So one question that we had in our minds is like, if the microbiome and the genome are both influencing a blood metabolite, is that overlapping in the variance explained, or is that additive, right? Is the microbiome doing something totally orthogonal to what the genome's doing? And so this is just an example of the top 20 or so hits. Generally speaking, if you just look at stacked models of the variance explained by microbiome and genome independently, the height of that bar is about the same height as the joint model when we include them all in the same model. So it does really seem to be the case that whatever the genome is doing to the blood metabolome is not talking to whatever the microbiome is doing. They're separate, and they can be used in aggregate to explain even more of the variation in what's going on in the blood. There was a slight but significant reduction in R squared in the joint models, but very, very tiny. I think it was the average was 0.003 R squared. We could also look at interaction effects between the genome and, between what your genotype was and a particular association between a metabolite and a microbe. So this is a cartoon kind of displaying that. So we might have a particular locus in the genome with the homozygous major allele, heterozygous and homozygous minor allele of people of these different genotypes. And you might look for a situation in which this association is significant and positive only in one genotypic background and not in the others. And in fact, we found something like 460 of these significant associations. This is just one example for a particular analyte called homoarginine, which is actually a risk factor for cardiovascular disease. So lower circulating homoarginine is associated with higher cardiovascular risk. This particular genetic locus is associated with increased risk for heart attacks. And you can see that it seems to bring down your homoarginine levels in blood. But we find that if you look at this analyte and this particular genus in the microbiome, having lower levels of this microbe in the gut of the homozygous minor allele population actually pushes their homoarginine level up to the wild type level in the blood. So it sort of indicates that you might be able to use this information to either mask genetic deficits or help people overcome their genetics, essentially. And I think I might be spending too long on this particular project, but I'll talk a little bit about this last point, where we were looking at, for metabolites that are in the blood, what determines whether they're under mostly host control or mostly microbiome control. And here we looked at secondary bile acids. So bile acids help us dissolve and absorb fats in our diet. We excrete them into the gut. They're toxic to bacteria, and bacteria chew them up with bile salt hydrolases and convert primary bile acids into secondary bile acids. And so you don't see secondary bile acids in germ-free organisms. They're exclusively produced by the microbiome. But they get absorbed into the blood, and they often get conjugated with little moieties in the liver, so they're more soluble in the bloodstream. So you get a glucuronide or a sulfate or something like that added to the bile acid. What we see is for unconjugated secondary bile acids, the variance explained in the blood is exclusively associated with the microbiome across all these different data sets we look at. But you see a flip towards having a large amount of the variance explained by the genome as soon as you add one of those moieties from the liver. So as soon as you sulfate or glucuronide these bile acids, the genotype takes control of the standing levels of these analytes in the bloodstream. And even the sort of associations with gut microbes, depending on whether you are, the same exact backbone of the bile acid, whether it's conjugated or not conjugated, drives differential associations with microbes in the gut. It's often thought that these conjugations can be food for microbes. They can actually cleave the conjugations and use them as a carbon and nitrogen source. And so they're either poison or they're food, and that can kind of flip around depending on these interactions. Okay, so that's sort of an overview of like, why is this important to think about the metabolism of the microbiome? Like, the majority of all the metabolites in our blood appear to be significantly associated with variation in the microbiome. So now I'm going to pivot to mechanistic modeling, this work that we're doing where we're trying to predict the response of someone to diet or probiotic interventions in a personalized way based on the metabolism of their microbiome. This little cartoon is an actual depiction of one of our genome scale models. So you can see the boundaries here are each an individual organism. These denser edges are sort of internal metabolic fluxes within that cell. And then edges across these density regions are cross-feeding interactions and metabolites being exchanged between microbes. And then some of these big clusters here are just sort of global metabolisms that are happening extracellularly, like bile acid metabolism and so on, that many things are contributing to at once. And if you're not familiar with flux balance analysis, I won't get too far into the weeds on it, but this is the basic textbook view. It's essentially taking a metabolic network. Since we can sequence an organism's genome, we can annotate all the enzymes, the metabolic enzymes it has. We can sort of see what it's capable of eating and producing from a perspective of metabolites. And if you were to try to build a dynamical model of these systems as differential equations, we're talking about hundreds of thousands of enzymes and reactions. And so it would actually take forever to try to fit that model. It's due to be too computationally intensive. So one way that people have made it tractable is to turn it into a linear algebra problem. So you convert a metabolic network into a matrix, essentially. So here's a little example of a network where you have reaction 1, 2, 3, and 4, and metabolites A, B, C, and D. And you build what's called a stoichiometric matrix, and it keeps track of the accounting of what metabolites are produced or consumed in a given reaction. So here reactions are columns and metabolites are rows. And you can see for example, reaction 2, it consumes metabolite A and it produces metabolite B. And so once you have this matrix, then the next question is, what is the velocity of material moving through any given reaction in the system? And that's what's called flux. So the basic equation is this S matrix is multiplied by this flux vector, and you assume a steady state in which the inputs to the system are balanced by the outputs. So in aggregate, everything is summing up to 0. And this allows you to essentially get a solution to this complicated question in finite time. You also can add a lot of constraints. This is often also called constraint-based modeling. So you can add thermodynamic information you have about the system and what conditions certain reactions occur under. This is an example of an allowable solution space over here, just looking at, say, 2 of these reactions. This space is where the S times V equals 0. So you take this infinite huge space of possible solutions and you collapse it down to a smaller but still infinite space of possible solutions. And then at some point you want to converge on a single solution, so you have to do something like maximize, minimize, or optimize in order to get to that solution. And so, for example, here, if we maximize flux through this reaction B5, we have a single convex solution here at this point. And if we do B6, our solution is here essentially. And these aren't 2-dimensional systems, right? They're high dimension. So here's this 3-dimensional version, and this is often referred to as a flux cone. But you have like a hyperflux cone in reality with like hundreds of thousands of dimensions, right? And you're trying to find a solution in that space. And for bacteria, the way you converge on a unique solution is often to assume maximum biomass production. So that's what you're maximizing. You're sort of assuming that the microbe is trying to grow as much as it can, as fast as it can on a given resource environment. Okay. And one of the inputs you need for these types of models are these genome-scale metabolic models that include all the enzymes that an organism has. It's stoichiometric matrix essentially is what you need for a given organism. And there happens to be a nicely curated database of these models called AGORA. And it's about 800 human gut microbes that have had whole genome sequencing done. And many of them have been cultured under many conditions to sort of validate the model predictions. So they've actually been experimentally curated in addition to being kind of computationally curated. So it's a high quality set of models. I think AGORA 2.0 is about to come out, and that's going to have like 7,000 or 8,000 models. And it's going to include organisms from other environments than just the human gut. So that should be useful to a lot more people. So what we've done in the last couple of years, and this is really pioneered by Christian Diener, this brilliant computational biologist in my group, is to extend flux balance analysis from single organisms. Like in the past, it's usually just been done to simulate E. coli growing in a bioreactor. And we're extending it to complex, diverse, multi-species communities. And so this platform we have is called MICOM, short for microbial community. Its inputs are the abundances of the various taxa in a given person's microbiome, which you can obtain through 16S or metagenomic sequencing. And you also need to provide initial conditions on the dietary inputs. We have a few that we've pre-computed, like the average Western diet or the Mediterranean diet, and so on. But you can also personalize dietary inputs if you know something about what someone's eating. It's a little tricky to convert, a banana into the sort of molecular components of what a banana is that the model can actually read, but it's doable. You can run these models. They give you a bunch of predictions like the growth rates of all the taxa in the system, who's producing what, who's cross-feeding who, what metabolites are being absorbed by the host, and so on. And how much time do I got? I should probably skip a little bit, but one of the things that was really a challenge with this type of modeling framework is that if you have these multi-species metabolic models, if you just do the naive thing and assume maximal biomass production, what ends up happening oftentimes is a single organism is growing at the end. You might have put in 100 things in there, but only one thing is left, like E. coli wins and everybody's dead, which is obviously not what we observe in vivo, right? So there's something wrong with the assumptions of the model. And just to give you an example, if you had two organisms, say two different E. coli strains in a system, there would be this sort of infinite set of solutions where you're achieving maximal community biomass production, but you could have it where one organism's growing and the other one's not growing at all, or vice versa, right? So we've implemented a few kind of ecologically grounded constraints to our modeling that allow us to achieve results that seem to mirror real data better. One is this L2 penalization that gives us kind of a convex solution in a case like this. And we can have a prior on this by inputting the abundances we observe in sequencing. And so the L2 sort of makes the growth rates converge more towards the observed abundance distribution. We can also allow the system to achieve less than the maximal community biomass. We can allow organisms to maximize their own biomass a little more at the expense of the entire community's biomass. And oftentimes you see this in invasive species and things like that, right? Sometimes more diverse communities actually achieve slightly less biomass. And we kind of see this in microbial communities as well. So, how do you gauge the success of a given model? One is whether or not you have things growing that are at appreciable abundances. You expect that the gut's like a live fast and die young environment, right? Is a constant flux of stuff going through. You're getting pushed out all the time. And if you're not growing, you go extinct. And so if the model's saying you're not growing, it's probably wrong. We find that, at a given trade-off level for this cooperative trade-off we're running, we tend to get this flip where most things are able to grow, and below that, things aren't able to grow as well. We can also independently calculate growth rates of organisms in microbiomes from metagenomes. So there's this phenomenon that you might be familiar with, where at the origin of replication, you often have to parallelize genome replication in fast-growing bacteria, because they can actually split their cells faster than they can replicate their genomes. So you can get many different replication forks in parallel going at once. And if you break up that bacterium and then you map all those short reads to its reference genome, you find there's a pileup of reads at the origin of replication, and fewer reads at the terminus. And the steepness of the slope has been validated as being very proportional to the rate of growth of that organism. So we can get these instantaneous growth rate estimates for metagenomes, and we find that at a certain tuning of our trade-off parameter, we can maximize the correlation between observed growth rates and model predictions. So what are the kind of things that the model spits out? These are some example plots from our paper that was the initial paper for the method. This in the left is an example of what is the microbiome eating. So we're feeding a bunch of people. These are, each column is a different human being. And we're feeding them all the standard Western diet in silico. We're giving them all the same in silico diet. And the colors here are the fluxes, the consumption fluxes of different metabolites that are in the diet. And so you can see there's different distributions here. So there's a group of people who tend to specialize more on degrading fiber. And then there's another group of folks that seem to specialize more on branched chain amino acid fermentation. And these are all, again, being fed the same exact diet. So their microbiome composition is driving very differential responses to that dietary input. We can also look at niche space in a very quantitative way. We can project this sort of consumption fluxes, like what is any individual microbe eating? We can project it into a ordination space. This is like I think this is like a Tisney. And so the coloring is coloring by genus, and you can see that all the little dots of the same color kind of cluster together, which means that those organisms, when they're growing in different people, have a similar metabolic niche, essentially. You can see which organisms are closer to each other in this niche space, and they tend to be maybe more competitive, or they have similar nutrient requirements. And things that are further apart in this space are more maybe complementary in their growth because they have sort of non-overlapping sets of nutrition. And I think practically speaking, from like a medical standpoint, one of the things we're interested with this modeling platform is it allows you to essentially do a bunch of experiments on a human being in silico that you couldn't do in real life. So this is an intervention analysis, and I don't expect you to read it all. It's too much information, but to give you some basic information, the top 3 rows are the fluxes of acetate production. The middle 3 are butyrate production, and the bottom 3 are propionate production. These are the major short chain fatty acids, or the end products of fermentation that microbes are producing in the gut. And the 3 rows in each cluster are 3 different individuals, 2 with type 2 diabetes and 1 healthy individual. And on the columns, these are interventions. So a dietary intervention in orange means we're turning up the abundance of just a single metabolite in the diet. Blue is a probiotic intervention where we can turn up the abundance of a single microbe in the ecological community and look at how that affects short chain fatty acid production. And if you zoom in on this one little region of the plot, you can see that there's a number of dietary interventions that can really juice up butyrate production in the healthy control, but you really see no effect in the 2 people with type 2 diabetes. So it potentially allows us to make precision nutrition predictions about how people will respond to dietary intervention. And so that's the question: can we use something like this to build a precision medicine or precision nutrition model that benefits people through leveraging the heterogeneity across them? And so the way we've been working on this is to try to show how quantitative our predictions can be under validation in validation experiments. So Nick Bowman, a student in the group, has been working on this. We pulled down one data set from Ophelia Venturelli's lab, where she's been culturing gut bugs in these tiny communities of 1 to up to 20 member communities and measuring short chain fatty acid production. And one thing we found in these really tiny in vitro communities is that the smaller the community is, the worse our model does. And we think this has something to do with the fact that our database models don't have the exact same gene complement as the strains that are actually in the samples that we're trying to model. And by including more taxa, you end up sort of masking those deficits in composition of the metabolic network, because you have more functional redundancy. And as the communities get more and more diverse, our predictions seem to get better. And we expect this to improve if we can start to get metagenomics and do our own assemblies and make our own models from a person's individual sample. But in lieu of that, leveraging the database models, we think that in diverse communities, the predictions, at least for short chain fatty acids, are working okay. We can get even more complicated than these in vitro communities by doing ex vivo experiments. And so this is a picture of poop soup, where we've taken a turd and we've homogenized it in a bag, and turn it into a slurry. We can put them in these deep well 96 well plates and add interventions to them. Like we can give inulin, for example, to some wells, which is the fiber in a banana, or we could do pectin or tryptophan or whatever we want. And then measure metabolomics over time and look at the production and consumption of these metabolites or fluxes. This is often called fluxomics. And then we can compare those fluxes directly to model predictions. And indeed, we kind of did that in a pilot study for a grant we were writing. And so this is just two people. This is with collaborator Joanna Lampe and Peter Dorstein. But we have two different stool donors. We can see that their microbiomes are different at the phylum level. You know, this person has more Bacteroidetes. And donor B makes more butyrate and more propionate than donor A on the exact same amount of inulin that you put into the slurry. And so the inulin production wasn't significantly different across the two, but B was making more of these short-chain fatty acids. You can ask, could MICOM have predicted this? And indeed, it seemed like it can. So for propionate and butyrate, we see that donor B is predicted to produce more. And acetate, there's really no difference in the prediction. But this was only two people, right? So we've kind of expanded it a bit and found a few data sets that were lying around out there in the literature. Just as reference, again, this is our data set in the control group where we just add PBS buffer to the incubations. You still get a little bit of residual SCFA production because poop still contains, you know, metabolites that the microbes are eating. They're not done yet, but you add more carbon and you get more production. And this GURI data set and this Cantu junglase data set, we can see, you know, we do get quantitative predictions, it seems, right? For these different interventions. I think this is pectin instead of inulin. And the GURI data set was a little messy. I think we have a lot of reasons to suspect that it's maybe not the best data set to use here, because oftentimes we see a non-overlap between the PBS control and the intervention, whereas in the GURI data set, they're actually pretty overlapping in their distribution for the measured amount of short-chain fatty acids. So we don't know what's going on there. But at least in this data set where we get good clearance between the intervention groups, we see decent predictions going on. So this is interesting and reassuring. We also wanted to look in vivo to see if we could explain heterogeneity that had not yet been explained by other researchers. So this is a study by Sonnenberg at Stanford, where they gave a bunch of people a high fiber diet. Generally speaking, people think high fiber diets should be good for you, or they should reduce your inflammation levels. But what they actually found when measuring a ton of different immune markers is there was a group of people who actually had higher inflammation at the end of the intervention, 10 weeks of fiber eating. And then two groups that did see a reduction in inflammation. And if we run those data sets through our prediction, through MICOM, we see that the high inflammation group actually is predicted to produce less butyrate and propionate than the other two groups. And in fact, strangely, if we have time series for these populations, the ability of the high inflammation group to produce these short-chain fatty acids declines over time during the intervention. And you know, we're still playing around with how you can apply these kinds of models, but this is an example of we can flip a population of 3,000 people where we have microbiome sequenced from a Western diet to a high fiber diet and see how that affects butyrate production, this beneficial anti-inflammatory metabolite. And indeed, you get increased butyrate on average on a high fiber diet, but there's some people who behave differently. So here's an example on the right-hand side here. We've colored the distribution by the quantiles, and you can see in this highest production butyrate group in the Western diet, if you project this purple distribution back onto the high fiber diet, some people actually decline in their butyrate production when you give them a high fiber diet. And so if you look at those folks, right, indeed you see that going from Western to high fiber, you get a crash in butyrate, which is counterintuitive. And then you can use the model to try to predict what interventions could rescue the phenotype, right? Could you bring them back up to where they should be? And here we found that if you just gave inulin as an additional input to the high fiber diet, you could actually get most of these folks increasing their butyrate levels. Okay, and I think I probably should wrap up soon. So I'll just race through this last example. So that was more on predicting functional outputs from a metabolomic standpoint. Can we predict relevant metabolites that are relevant to human health?
You've got 10 minutes.
Oh, do I want to have time for questions, I guess.
Okay, 5 minutes.
Okay, that's good.
10 is fine.
Okay. All right. I won't rush too fast then. So this is another application of our modeling platform, which is kind of an ecosystem engineering application. Oftentimes people take probiotics, and the probiotic sometimes sticks in a person, or sometimes it doesn't. And if you stop taking that probiotic, it crashes out of your system. And how do we know when something will stick or when it won't stick? And so we think we can kind of get at that with this modeling platform. And Alex Carr, a grad student in my group, is working on applying this to Clostridioides difficile, or C. diff, which is it's an opportunistic pathogen, but up to like 30 or 40% of us carry it around asymptomatically as just a normal commensal. So it's sitting there lying in wait for us to get sick and have antibiotics or whatever, and then maybe it erupts into disease. But we wanted to know, like, if you expose someone to this organism who didn't already have it in their system, what are the chances it would stick? And so we're calling it in silico invasion assays, where essentially Alex is injecting 10% abundance C. diff into the metabolic models of people, running the model back to equilibrium and seeing, which folks are susceptible, where the predicted growth rate of C. diff is high and positive when it's injected into the model, and what folks are resistant, where the predicted growth rate is essentially 0 for C. diff, and then looking at the metabolic signatures of those ecosystems. So, again, proof, kind of sanity checks or proof of concepts here. There was a data set from my postdoc lab. This is from Eric Allen's group, where they tracked people over time series of stool microbiomes. And in this particular donor, there was a food poisoning event. This person had I believe it was a piece of French toast, and they got a salmonella infection. And that's what this red pulse is. And a follow-up analysis found that at that point, even though it was a salmonella infection, they were colonized by C. diff. That's what this blue kind of trace is here. But from 16S data, they found C. diff was colonizing this person after that event. This is kind of well known. If there's a disruption in your microbiome, it's often a window of opportunity for things like this to happen. And we wanted to ask whether MICOM could show us something similar. So we tried to inject 10% C. diff into this person's model throughout the whole time series, and that's what the green line is showing, is the growth rate of C. diff. And we find that we can't get positive growth until this point, and then suddenly, boom, we can like patchily get prediction of growth in this person's system. So this was sort of a nice qualitative validation. There was another time series where that person kind of had C. diff at a low level, kind of at the detection limits. That's why you see so many spikes. It's like 0.0002 relative abundance. But they were colonized throughout the time series, and we also show kind of patchy prediction of growth throughout the time series in this person. We can also look at another data set of FMT recipients. So here we're looking at the predicted growth rate of C. diff in the microbiomes of different folks in patients pre-FMT. So they've just gone through a bunch of antibiotic treatment, and they have C. diff, and they're about to get an FMT, and they're overwhelmingly more likely to have a strong predicted growth rate for C. diff. The healthy population is kind of bimodal, right? You see that some people, C. diff can grow in their system, and then some people it can't. And we see that post-FMT, the population sort of mirrors what you see in healthy people. We even had time series for a lot of these people. So these are 4 different recipients who received FMTs, and the circle point is where they started off, and the star point is where they end up. The ordination space is the sort of a metabolic import flux space for C. diff when it's growing in the system. And the heat coloring is the growth rate of C. diff. So darker color is lower growth rate and lighter color is higher growth rate. So often people are starting off in this really high predicted growth rate region. They bounce around a lot after their FMT, and they often end up in this low growth rate region of the metabolic space. With the exception of, I think, donor CD3, who starts off here in a high growth region and like ends up here in another high growth region. But we kind of know that this is the case. People can get recurrent infections all the time, even after an FMT. And if we look into the like metabolic details of what's going on, we see some interesting stuff. So for example, C. diff appears to have multiple metabolic strategies depending on who it's living in. So there's the top red bar here is the growth rate of C. diff. So darker red is higher growth and light is 0 to no growth. And there's a really high growth rate cluster here where it's importing a lot of these metabolites, that many of which have been shown empirically to be important for C. diff growth, like succinate and ornithine and different amino acids involved in Strickland fermentation. But there's another growth niche, it appears, where there's a bunch of metabolites that are not consumed. And even so, C. diff is still growing pretty fast. So for example, in this growth niche, ornithine isn't really being consumed, but it's being consumed at a high rate in this growth niche. So C. diff has this kind of flexible plasticity to its metabolic capacity. Its sort of realized niche can be kind of different from person to person. And then if we look at heat maps here now of production and consumption fluxes for some of these important metabolites that are involved in C. diff growth, I think the top row is import. So it's what these things are eating. And the bottom row is export. So it's what they're producing. Oops. And you can see that, Clostridioides is importing a lot of succinate, and as you expect. And there are some other organisms that seem to be keep competing for succinate. So Coca-Cola here, it's importing a lot of succinate when C. diff is not importing a lot of succinate. So we can maybe identify competitors with these import fluxes. And with export fluxes, we can identify facilitators. So for C. diff here, it's when it's growing fast, there's a lot of organisms that are kind of clustridial in nature that are producing a lot of succinate. And it looks like they're cross-feeding C. diff. And so we're kind of digging more and more into this. We're trying to write this up right now. Again, this is like the multiple growth niche, but in a dimensional reduction plot across different data sets. We want to know whether these differential niches are conserved across many data sets. So this is the Elm data set, the FMT data set, but then this is another data set we have access to, the Arabelle data set that I talked about earlier, and then the American gut data set, which my student humorously called Murica. But yeah, you have, it seems the same set of clusters, right? Really high growth cluster, an intermediate growth cluster, and then a cluster of essentially 0 growth. Another, gosh, I think I need to wrap up, but yeah, another quick observation is there seems to be a Goldilocks zone for alpha diversity. So at really high diversity, we see higher C. diff growth, and at low diversity, we see higher C. diff growth, but intermediate levels of diversity, we get a sort of average the lowest predicted growth rate across people. You can look at C. diff's niche in the context of the growth niches of all the other taxa in the system, and we can see that it really does have these really divergent metabolic niches, it seems, within the context of a community. And you can look at that in the context of other organisms as well, like some of the ones that seem to be cross-feeding it, like Eubacterium and Faecalibacterium and Blauschia. And finally, we can see signatures in the blood that are associated with this predicted growth rate. So these are blood metabolomics and clinical labs that appear to be associated with the predicted growth rate of C. diff. And we can explain about 5% of the variance in C. diff growth rate from the model using this set of blood metabolites. So yeah, I hope I gave you a picture of the kind of stuff we're working on and sort of this vision we have. This is actually the title of a grant we just got funded. This vision we have of trying to build a mechanistic model that allows you to essentially build a digital twin of an individual person and their gut microbiome and their metabolism in silico, and do things to that and make predictions for interventions that you can maybe carry out in a trial. With that, thanks to everyone involved in the work. Thanks to my lab, really fun group of people, and the funders. And thanks to you all for listening.
Wow, I loved all of those great pictures.
Thank you.
Do you want to do I'm going to turn on this thing, and could you take around the microphone for the...
Have any questions?
People on Zoom have questions, and type them in or raise your hand. Yeah. That's
Okay,
fine.
cool.
You can hear us, right, John? Yes.
Yes.
You can hear us.
Yes. Yes. Oop. see a Zoom raised hand.
Zoom first.
Zoom first, and then Brendan has a question. Oh, Stillian, do you want to give your question?
Oh, sure. Thanks. Thanks, Sean, for this great talk. I really love your work. Very, very bold and ambitious, I have to say. So I'm really anxious to see what comes out of this in the long term. I had one question regarding your CTFBA framework that you introduced. So my understanding is that traditional FBA, the principle of optimization of biomass production rate, is justified by the idea that evolution selects for intracellular regulatory mechanisms that optimize growth. But this assumption cannot really be extrapolated to the community level, at least not based on classical evolutionary theory. So unless you assume group selection, which you haven't explicitly mentioned, and even if you do assume group selection, which is debatable, how can that still hold once you start introducing new strains that haven't co-evolved with the community?
Yeah, and that's really great. Thanks for that. Honestly, that's our critique of the field, because if you look at others who do this work, this is what they do. They assume that you're maximizing the community biomass, but I agree that there's really no evolutionary mechanism for why that should be the case. And in fact, every organism in the system should be maximizing its own biomass. There's more selfishness going on. But the problem is, if you have this multi-optimization model with hundreds of species, it's intractable to fit the model, right? And so almost everyone else who does this type of work just assumes that total community optimization, which I think is fundamentally wrong. We're gonna, I think, write a perspective piece about this in a bit. But yeah, we try to have some heuristic approach that sort of straddles in between having a fully multi-optimized model and having some basic rules you can put in place that allow for individual taxa to optimize their own biomass more, and not focus exclusively on community biomass as the optimization. But it's a great observation.
Solutions that actually are below the community optimum in principle.
Yes, that's part of our framework, is we allow the system to achieve less than the community maximal growth rate, allowing other taxa to achieve higher growth rates.
Thanks.
Yeah.
Hi, Sean. Can you hear me?
Yes.
That was a great talk. It was really fun. I have 1,000 questions, but I want to focus on the C. diff modeling, which I think is a great example of kind of bringing together invasion ecology and pathogen biology to help us understand what drives pathogen invasion. So I had two questions, a little one and a bigger one. The little one was, I was really intrigued by the Goldilocks result with alpha diversity and invasion success. And what do you think underlies that particular example? And then I have another question.
It's a good question. At a more like host phenotype level, we have definitely observed that, if you have really, really low diversity, if I looked at who that person was, it's very likely that they're either sick or they've taken a lot of antibiotics, or they have a really low fiber diet. And then often people who have very, very high diversity, they're kind of outliers on the high diversity side, they're often constipated. And so they're sick in their own right, and the system is sort of out of whack. So we know like functionally the host is a little bit out of whack on the fringes of the distribution. Why does that allow C. diff to grow better? We don't quite understand yet. We're trying to get at that. I think it's something to do with the fact that by having more taxa around, there's sort of this interesting fractal nature to microbial communities where the more diversity you have in the system, the more niches exist because of all the metabolic byproducts being produced by these other taxa. And so in really, really diverse systems, it's hard not to grow, I think, sometimes, because even though you get saturation of some of the larger niches, you actually construct a bunch of sub niches that are smaller, but they just sort of explode with more diversity.
Great, thanks. So my other question was just, given all of the different ways you've explored invasion success with that model, are there general rules that you've kind of can establish now for what allows invasion success in the gut?
Yes. So general rules. Yeah. I mean, we definitely see that the organisms, the metabolites that appear to be driving this. So, if your microbiome is allowing for a lot of excess succinate to just sit out there in the lumen, or a lot of ornithine to build up, there's a good chance you're going to get invaded by C. diff. And certain taxa seem to contribute to that more than others. And so composition can tell you something, but then the modeling tells you which metabolites are seemingly responsible.
Great, thanks.
Yeah.
Any more questions in here or on Zoom? Any Zoom questions?
I was wondering, what else would you need to know to further constrain your, further constrain your solutions so that it doesn't depend so much on regularization or these maximizing growth assumptions?
Good question.
That's plausible, we might know. Yeah.
So one way you could do it is to essentially use what I referred to, these peak to trough ratios or sort of instantaneous estimates of growth rate directly from metagenomes. You could directly use those growth rate estimates as the constraints on the growth rates of the taxa in the system without fitting the growth rates as much, right? So that's one thing one could do. So far we've only used the growth rates as a validation to ask, whether the models are predicting things that seem to be correlated with reality, but you could just bake those inferred growth rates from a metagenome directly into the model. So that's one way to get around the CTFBA, some steps in the CTFBA. But it requires having metagenomes, which not everybody has.
Yeah. Stilling, you have another question?
Yeah, I have another question about the earlier experiment that you started with, where you separated the variants explained into the microbiome versus the genome of the host. And perhaps I didn't understand the experimental design completely, so perhaps it's just a matter of elaborating that. But where does the environmental factor get absorbed in? So things like diet and behavior of the host, or were those completely controlled for?
So we only controlled for a set of covariates we tend to control for, like BMI, sex, age, and we did the 5 first principal components of genetic ancestry, or whatever you'd call that, ancestry, I guess. But for diet and all of that other stuff, sadly we didn't have that information for this cohort. I expect that a certain fraction of the variation of diet is going to be bound up with the variation of the microbiome. So they're kind of co-correlated, but there's also going to be more variance there that could explain. I mean, in fact, there was a paper that came out in Nature Medicine actually around the same time as ours is coming out that had diet in addition, and they show some associations there that are independent of the microbiome. So that's definitely a part of the picture. From a modeling standpoint, all that variation just went into the residual, right? So we're not capturing it.
Okay.
Any more questions?
Thanks, Sean. Is there a thing
Yes, there is tea time on the same Zoom link starting now, I guess, in